Hi everyone, my name's Alan. I'll be presenting here today, the last one. So a little bit about myself. I'm currently a second-year student at UIC in the Department of Pharmacognosy, which is the drug discovery from natural products. I did my undergrad at Northwestern. I graduated 2010. I worked 5 years in a small company called Botanical in Connecticut, which is specialized in chromatography, such as countercurrent chromatography, ion exchange, and so on, to isolate phytochemicals. And then we send them off to a research group around the world to do the research. And I got interested in working with plants and mostly isolating compounds from it. That's how I got into wanting to go back to school again. And now I'm in UIC doing my second year. I'm in Dr. Ben Breeman's group at UIC. And we're kind of like the mass spec lab in the college. So there'll be a lot of mass spec. So our lab is part of the UIC Botanical Center, which focuses on women's health. Our lab is the analytical core, which does the analysis for the clinical studies. There's also another lab which does the plant authentication, and another lab which does the bioassays. We've been funded by the NIH for 19 years now, 18 actually. And so we focus on the safety and the mechanism of action of botanicals that women use as alternative to hormone therapy. And what you see here on the slide is, I can't see where I'm pointing to. Oh, okay. Over here is the sales of botanicals in the US in the last 10 years. And last year, there was the sale just in the US was $7.5 billion. And it continues to increase about 7% every year. And I mean, that means a lot of people are taking botanicals, but how effective are they? But in our recent years, we're actually more focused on looking at drug-botanical interactions. And what that means is, well, in our human body, we have a lot of enzymes which metabolize like our own endogenous compounds, as well as xenobiotics, such as drugs and these botanicals. But surprisingly, this is the metabolism of xenobiotics. Most of the drugs we take are metabolized. And of the metabolism, which is the purple here, it's really just a handful of enzymes, like 20 of them. And when we take botanicals, it actually affects the regulation of these enzymes. So, like for example, when you take meds, your doctor might tell you, don't drink grapefruit juice, because grapefruit juice actually is an inhibitor of cytochrome 3A4, which, and you see here, it actually does like half of the metabolism of the cytochromes for your drugs. So it actually affects a lot about what you're taking. And sorry. So my project in particular is looking at botanical effects on estrogen metabolism in the body and how it affects, because estrogen, as you'll see, can actually cause cancer. So I'll go a little bit into the biology. I'm not a biologist, so I'll kind of be brief about this. So estrogen has two main metabolism pathways. The first pathway is the hydroxylation on the 2 position, which is also called the nontoxic pathway. It's relatively safe, and after hydroxylation, it gets methoxylated and eliminated from the body. And the other pathway is hydroxylation on the 4 position on the A ring. And this can actually be oxidized to a quinone. And this quinone here can form a DNA adduct. And with reactive oxygen species can cause mutations in your DNA. And this COMT, catechol-O-methyl transferase, actually helps detoxify this step by eliminating these 4-hydroxy estrogens. And how botanicals come into play is they regulate all these enzymes. So the botanicals can attach onto NF-kappa B, which is the inflammatory pathway, which inhibits the cytochrome 1b1. And so you get less of these 4-hydroxyl catechol. You can also have, it also works in a chemopreventive pathway, which it activates the KEAP1 and NRF2, and it upregulates the arrow response element in your DNA. And it will increase, upregulate the NQO1, which helps reduce these quinones back to their 4-hydroxy catechols. And in addition, phytoestrogens from botanicals can also bind to ER receptors, estrogen receptors, and that could actually lead to proliferation of cells, which would be bad in terms of if you have cancer. So there's a estrogen quantitation has been around. People have been trying this for over 10 years. And there's a number of challenges with this. First of all, these estrogens are in very low concentration. They're in picogram per ml. The catechols would be around 1 or 2 picogram per ml. In most of these studies, they actually marked as below limit of detection. And the reason for this is because these estrogens have very poor ionization. The ionization, the most acidic group is actually this OH over here. And it has a pK of 9.5, 10.5. And if you compare it to a lot of other compounds in the biological matrix, such as amino acids and phospholipids, which have a pK of around 2, they are much more difficult to ionize. And these are some of the approaches that people have taken in the past. Actually, I'll take one step back. A lot of people, aside from these attachment chemical modifications, people actually try to look into it just by negative mode LC-MS-MS, such as with ammonium fluoride additive. And ammonium fluoride, as you know, it becomes HF in your mass spec, which is terrible for it. And you also get HF waste, which you don't want to deal with. But that's the approach people are taking for wastewater measurement of estrogens. And for looking at biological samples, they want to modify it. So danzolation is the first of the modifications that's been published in 2007. And it's by using a sulfonochloride group to attach on the free hydroxyl group. And similarly, you also have these pyridinium sulfonochloride and some other ones which uses the sulfonochloride group. There are other ones such as the pyridinium, fluoropyridinium, or the acid chlorides to attach to the estrogens, but these are very non-selective and you get quite messy reaction. And lastly, you also have Gerard P reagent, which modifies the estrone, but this the ketone, but this only works on estrone and not estradiol, which is the other form of estrogen. So what I'm looking at using is pyridinium sulfonochloride derivatization, but there's a second step, which is the N-methylation, and this is the estrone. And in your body, it's usually excreted as a glucuronide or a sulfate conjugation, which is a phase 2 metabolite. And to deconjugate it, it's an incubation of around 20 hours with beta-glucuronidase and sulfatase to remove this. And after liquid-liquid extraction, you also, with MTBE, you remove the proteins from the biological serum samples, in this case, human serum. And then you can do pyridinium sulfonochloride incubation at pH 9 for 30 minutes to attach this group onto the OH. And after that, you need to dry it down again with another MTBE extraction, and then with N-methylation with iodomethane for another 30 minutes at a higher temperature at 80 degrees to add a methyl group to get a positive charge on the nitrogen. And it's actually the same reaction, this first part, as what's been described in the literature. But the second part is actually very important. As you'll see, And these are the fermentations. As you'll see in the mass spec over here, I'll go a little bit into the background first. So for mass spec, I'm using Shimatsu triple quad LC-MS 8060, and using MS-MS selective reaction monitoring for better signal to noise, so I can do the quantitation in a more sensitive manner. And for the non-derivative estrogen, I look at it in negative mode, and with just water to acetonitrile from 25% to 40%. And then it's 100% acetonitrile wash, followed by a 0% water wash, and then equilibration. And similarly for the NMPS derivation, but this time with 0.1% formic acid added to the water and run in positive mode. And for the ionization part, we have the capillary tube, and it forms a Taylor cone from the pole of the counter electrode. And from here, the counter electrode pushes the negative charge ions back into the tube, but the positive charge gets pulled out and eventually breaks up into droplets. And the droplets have charge in it and the molecules, the compounds, the analytes. And with the drying gas, the solvent evaporates, but the charge remains, and eventually you get a Coulombic fission where when you have like too little solvents and too much charge, so it kind of spreads out, it breaks off into like smaller droplets. And then it either goes through field-assisted desorption or just charged residue model, where in charged residue model, the solvent just evaporates until you have just analyte, and field-assisted ion desorption is the analyte gets ejected out and carries the charge with it. And this is how the molecule gets into the mass spec. So what's interesting about having a preformed cation is that you already have a charge on the molecule. You don't need to ionize it. So in this case, I tried many different ionization potentials. So at very high, I tried 5,000 volts, and you actually have a very small signal. And the reason for that is because it's in-source fragmentation. You're losing your signal because you're overcharging your molecules. And when you go lower down to 0.1 or 0, you actually get a very low signal or a very distorted peak. And that's because the Taylor cone over here isn't formed properly when you don't have enough ionization energy. So we found that the optimal for these pre-charged cations is around 0.5 kilovolts. And so how does that impact what we're looking at? So for estrogens, without the derivatization in water, you see all these peaks. These are the hydroxy compounds, 2-hydroxy, 4-hydroxy catechols. And when they're in serum, you actually don't see any of them. It's because of matrix effects that the other biological molecules in the serum are actually interfering with the ionization. And when you actually lower the ionization potential, because you have these for these derivatives, you don't lose any of the signal. But one downside to this derivatization is for these 2-hydroxy and 4-hydroxy estrogens, they actually have 2 sites that you can attach the pyridinium sulfate to. And each of them, you can have the iodomethane attached here, here, or on both of them. So in a sense, you get 3 metabolites instead of 1 to quantitate. And luckily, so this is run with just E1, estrone for methoxy estrone, which only has 1 OH group, so you only get 1 compound. And the 2 pink peaks over here are these 2. One is over here, one is with the charge on over here, and one is over here. And the reason why they're separated is because the chromatography is actually able to tell them apart. So they come off at different times from the column. And what's most interesting about this is this giant peak over here, which is the 2-charge molecule. And surprisingly, it gives a much stronger signal than everything else, even though they're at the same concentration. And I don't know the reason for that, but a postdoc in our lab who does a lot of proteomics often claims that the double charged molecules just fly better in the mass spec. So I will take her word for this. And when I combine all 10 of the metabolites together running through mass spec, I didn't label all of them, but you can actually tell them all apart because they have different SRM transitions. And for the ones that are isomers of each other, they separate quite well. The only difficulty now is these double charged molecules, they tend to come off the column at the same time. And thanks to Rich, I've actually been testing a lot of different columns from Waters and Thermo, who've been helping me out. And from all the columns that I've tested the first 5 over here. I have the best result from the fluorophenol column and the T3 columns. And I haven't tested these 2 yet. This one is a C18 embedded with weak cation exchange molecules, by the way. And one other thing I want to modify is to work on is the reaction conditions. By optimizing the reaction time and temperature, I hope to push the reaction to completion to get just the 2 charged molecule and none of the 1 charge hydroxy molecules. And also I want to work, this is something I'm working on right now, is doing the second step derivation with isotope label methyl and this will help me determine what the fragments are when I run the SRM to determine where the fragmentation happens. And I hope to do this with the Waters G1 QTOF in the next week or so. And I want to try out alternative reactions. The GRP, which I mentioned before, it targets just the ketone site. Although this only works with like half of the molecules, but it will be a very simple reaction since it's only 1 step instead of 2 step. And these 3 over here are ideas that I came across. These are studies done in the 1960s, and I haven't found anything more recent than that. But estrogen can actually form a conjugation to amino acids, to urea, and to caffeine. And I need to find the exact enzymes that does these, but hopefully I can. It'll be like a different direction to take this project in the near future. In addition, currently we're working with serum because we have serum from these clinical trial from black cohosh, from hops, from red clover, and also from a control group taking Prempro. But we also have urine samples, which should have a lot of the catechol estrogens. So that would be a much easier source to work with, and it's also more abundant. And I'm also working on modifying the LC system, because I've done this before, but not here in this lab. So by hooking up a second trapping column with weak cation exchange, I hope to inject a much larger sample size so I can improve on the quantitation, like lower limit of detection. Because right now, my limit of detection is around 5 picogram per ml, and I hope to get it down another magnitude, because a lot of these compounds are around like 1 picogram or 2 picogram, and I'm just above that a little bit right now. And I also want to work with some other ion pair agent. Maybe this, if I use this stronger, I'll improve on the chromatography, but it'll also like negatively impact the mass spec. So it's kind of a trade-off. So I just have to find the right one. Thank you for listening, and I want to